So we were talking about the 5 prime UTR and the 3 prime UTR, and I have said you an example based on this 5 prime UTR that there are special RNA binding proteins that binds to this UTR sequence and prevent the and regulate the translation process, post transcriptional modification process. So one of the best example is your MSL2 gene. So I am getting discussing with this part. What is MSL2 gene? MSL2 is actually a protein coding gene, and it is a component of histone acetyl transferase complex. MSL2, it is a protein coding gene, and it is a component of your histone acetyl transferase complex, which is responsible for the histone acetylation of histone H4 at lysine 16. Means from the N terminal of histone H4 there is a tail. From that N terminal, if you move on towards the C terminal, at the 16th amino acid is your lysine, that is getting acetylated by this enzyme, by this product of MSL2 gene. It is a component of histone acetyl transferase complex. It performs acetylation of lysine at position 16 of histone H4, right? Now it has many diverse function also. This MSL2 gene also act as a E3 ubiquitin ligase. It also acts as a E3 ubiquitin ligase that will promote monoubiquitination of histone H2B at lysine 35. What is ubiquitination? In ubiquitination, normally a protein is activated by attaching ubiquitin into it. Okay, in ubiquitination, a protein is activated by attaching ubiquitin into it. Ubiquitin is a small molecule which acts as a tag. It acts as a tag, a mark that will signals the protein transport machinery to transport the protein into the proteasome. Normally, ubiquitin is a small molecule which acts as a tag which signals the protein transport machinery to transport that protein into proteasome, where it will be further degraded, where they will be further degraded by proteolysis. Proteolysis is a chemical reaction which will break the peptide bond. So this MSL2 gene also act as a ubiquitin ligase which will promote ubiquitination of H2B. It promotes ubiquitination of H2B at lysine position number 35. Okay. Now this activity of ubiquitination promoted by this MSL2 gene is greatly enhanced by heterodimerization with MSL1. MSL2 gene alone cannot the product of MSL2 gene alone cannot do this ubiquitination. With the help of this MSL1, they form a heterodimer, and that leads to together they enhance that ubiquitination of H2B at lysine 35. Okay, now this ubiquitination of H2B will then stimulates your histone H3 methylation. Okay, H2 ubiquitination, H2B ubiquitination as done by these MSL2 and MSL1 gene product, This H2B ubiquitination will stimulate your histone H3 methylation, okay, at certain regions. Histone H3 methylation will be then carried out at lysine position number 4 and 79. That leads to gene activation of your important genes like your HOX A9, then MEIS1 gene. So it is a regulatory gene, right, this MSL2. Now it has also been shown about the SXL protein. SXL is an RNA binding protein. What does it do? The full form of SXL is sex lethal. It is an important post-transcriptional regulator of gene expression in female Drosophila. This SXL gene, it is an RNA binding protein. It encodes an RNA binding protein, which is an important post-transcriptional regulator of the gene expression in female Drosophila. Okay, and this SXL acts as a repressor of the MSL to mRNA translation. This SXL protein will act as a repressor of the MSL to mRNA translation. How? It has been shown. Now let us look at the written material. It is showing that the protein SXL attaching to an intron segment located within the 3' UTR segment of the primary transcript. So within the MSL2 gene there are intron regions, exon regions. SXL bind to the 5' untranslated region which is present on within a intron on the upstream, and due to attachment of SXL to this 5' UTR region of that intron found in MSL2 gene, it leads to including that intron even after processing. It includes that intron. Normally introns are getting spliced out after processing, after splicing process, but this intron will remain as a result of alternative splicing, actually you can say. So SXL protein is a RNA binding protein. It interacts with the MSL2 gene. How? Within the MSL2 mRNA there is a 5' UTR segment, right? Here is a 5' UTR segment in one of their introns. SXL binds to that 5' UTR region of that intron and leads to include that intron even after processing. The intron remain in that mRNA, and this will allow recruitment of the proteins which will bind simultaneously to both 5' UTR and 3' UTR. This will allow recruitment of the proteins which will bind simultaneously to both 5' and 3' UTR, not allowing the translation proteins to assemble. However, okay, so simple thing. So SXL interacts with the MSL2 gene. How it interacts with the MSL2 gene? How it regulates the MSL2 gene? It binds to the 5' UTR of MSL2 mRNA. The 5' UTR is located in one of their introns. It binds and it leads to including that introns even after processing. Inclusion of intron within the mRNA still after processing. Due to the remaining of intron in that mRNA, other proteins binds simultaneously to both the 5' and 3' untranslated regions. okay, and thus it prevents the translation proteins to assemble. So this is actually a type of regulation done by this RNA binding protein as done by SXL. Okay, now I will talk you to a next example that has not been cleared perfectly in the last video, that is your this one. I have also said that another important regulation of translation is interaction between the untranslated regions of 3 prime and 5 prime. So the 3 prime UTR and 5 prime UTR, they interact with each other to form a circular mRNA which will regulate the translation process. Okay, so this has been observed in especially in Xenopas laevis, which is a type of frog. Okay, normally what it do, let us see. It is saying that EIF4E, which is a eukaryotic translation initiation factor, EIF4E bind to the 5 prime cap and it interacts with the masking bound to CPEB on the 3 prime UTR. What is masking? Masking is a protein factor. It is a CPEB associated protein factor. That means this masking is a type of protein factor which is associated with CPEB. Now what is CPEB? CPEB is cytoplasmic polyadenylation element binding protein. CPEB is a cytoplasmic polyadenylation element binding protein. This CPEB actually interacts with the CPE element found on the 3 prime UTR. CPEB, cytoplasmic polyadenylation element binding protein, interacts with the CPE element located on the 3 prime UTR, and they are with the CPEB masking protein factor bounds with CPEB, and thus creating a translationally inactive transcripts. So the EIF4E interacts with the 5 prime cap. Sorry, EIF4E bound to the 5 prime cap, and this EIF4E bounded with 5 prime cap, now it interacts with the CPEB on the 3 prime UTR. So it interacts means it associates now. It forms it interacts to bind together and forms a circle, mRNA, a circular mRNA, right? So EIF4E interacts with the CPEB, which is bounded by to the 3 prime UTR, and thus inactivating the translation machinery. Okay, now it can be again inactivate. This inactivation of translation can again be activated. How? If we do phosphorylation to this CPEB on the 3 prime UTR. So if CPEB is phosphorylated, it will getting displaced. The masking binding site where it binds with the CPEB, it displaces, thus allowing polymerization of the poly A tail, which can be recruited by the translational machinery by means of poly A binding protein. So CPEB is the main cause. It binds with the 3 prime UTR on the CPE element, and the CPEB interacts with the masking protein, which is a type of associated protein factor of CPEB, this masking, and then it interacts with the EIF4E, which has been attached with the 5 prime cap, and due to binding with the 5 prime cap, it forms a closed loop like structure. Okay, and thus translational machinery cannot able to come and bind and carry out translation. But this can be prevented also. How? If we do phosphorylation to this CPEB, then due to phosphorylation of CPEB, it will displace the masking binding site. It will displace the masking binding site, and thus allowing polymerization of the poly-A tail as done by the some polymerase, and then it will include the translational machinery to begin their translation synthesis. Okay, and this poly A tail is also covered with a protein called poly A binding protein. It will help to do that translation process efficiently. Okay, so this was two things that I have not discussed in the last video. Okay, coming to the next thing, your 3' UTR, the 3' untranslated region. So I have also discussed about the 3' UTR when we are talking about 5' UTR. The 3' UTR is present just after the stop codon, followed by a stop codon, right? And then you have the poly A tail. So first there is gap, then your 5' UTR. These 5' UTR are untranslated regions. They are not getting translated actually. This 5' UTR also contains what? They also consist of UORF, as I have mentioned you before. This UORF acts as a regulation of translation. Within this 5' UTR, some regulatory proteins, RNA binding proteins, binds and regulate the translation process. Then comes your coding sequence, the ORF, the open reading frame. Here start and stop has been written. That means here from there start codon starts and the stop codon, it will terminate the translation. Then comes your 3' UTR, and finally your poly A tail. Okay, normally 3' UTR is about 700 nucleotides long. So in molecular genetics, the 3' untranslated region is the section of mRNA. The 3' untranslated region is the section of mRNA which is immediately follows the translation termination codon. The 3' UTR is located just after your termination codon. The 3' UTR often contain regulatory regions that are post-transcriptionally influence gene expression. Okay, one of them is your CPE element that I have shown. I have just now I have said to you, as found in Xenopus laevis. So 3' UTR often contains regulatory regions where your some regulatory proteins bind, and it influences the post-transcriptionally expression of gene. During gene expression, an mRNA molecule has to be transcribed from DNA sequence, and it is later translated into protein, that you know from the beginning. Several regions of the mRNA molecule are not translated into a protein, including a 5' cap, the untranslated region at the 5' end, that is the 5' UTR, the 3' untranslated region, that is 3' UTR, and poly A tail. So these are the regions which are not getting translated in mRNA. That is your the cap. The cap will not get translated. It is also acts as a recognition site for the ribosome. Then comes the 5' UTR, the 5' untranslated region. It is not getting translated. Exceptional in some case, for regulation it get translated, but it is not common. Then comes 3' UTR, the 3' untranslated region, and a poly A tail. Regulatory regions within the 3' untranslated region can influence your polyadenylation. It will include your translational efficiency, localization, and stability of mRNA. The within the 3' untranslated regions there are some regulatory regions, as I have said just now, and these regulatory regions can regulate your polyadenylation form. Then it will regulate your translational efficiency, the localization, and the stability of mRNA. The 3' UTR contains both binding sites for regulatory proteins as well as for microRNA, which is also called miRNA. MicroRNA and another RNA is called siRNA, short interfering RNA. They involve in a process called RNA interference, which actually able to regulate the transcription process, right, and also translation process. Especially for the translation process it regulates. So again I repeat, the 3' UTR contains the binding site for the regulatory proteins to as to regulate during the translation, as well as they recruit the miRNA or microRNA. MicroRNA are short RNA which are actually prepared by certain enzymes known as dicer enzymes, DICER, which we will talking you elaborately in the next video. Now, by binding to these specific sites, when this 3' UTR is interacted by these regulatory proteins, mRNA can decrease the gene expression of various mRNA by inhibiting the translation or directly causing degradation of the transcript. So, by binding to specific sites, when these 3' UTR having some regulatory regions where your regulatory proteins will bind, as well as your microRNA, siRNA will also bind. By binding by binding of these regulatory proteins to the 3' UTR, it will regulate your gene expression. It will decrease the gene expression of other mRNA. How? By inhibiting the translational machinery to bind or directly causing degradation of that mRNA. Okay, coming to the next image, this 3' UTR also has a silencer region where a repressor can bind and inhibit the expression of mRNA. This 3' UTR has a silencer region which binds to the repressor proteins. Within the silencer region, repressor protein will interact and will inhibit the expression of that mRNA. Many 3' UTR also contain AU-rich elements. Many 3' untranslated regions also contain AU-rich elements. In short, it is called ARE. Proteins that bind to these AU-rich elements also affect the stability or decay rate of the transcripts in a localized manner or affect the translation initiation. So the proteins that binds to these AU-rich elements located within the 3' UTR mostly, they can affect the stability or decay rate of the transcripts in a localized manner or affect the translation initiation. Furthermore, 3' UTR contain the sequence of AAUAAA, AAU AAA, which will direct the addition of several adenine residues called poly-A tail. This 3' UTR followed by a poly-A tail, right? We have seen the image, the 3' UTR followed by a poly-A tail. So how it is recruited? Actually, the 3' UTR have this sequence. They have this sequence AU, AAU, AAA, which will direct the addition of adenine residues called poly-A tail done by the poly-A polymerase. to the end of mRNA. Poly A binding protein called PABP bind to this poly A tail to contribute the regulation of mRNA translation, stability and export. There is also another protein called poly A binding protein which binds to this tail and contribute to the regulation of mRNA translation, stability and export. For example, how this poly A tail binding protein causes the regulation of translation and control the mRNA export. Let us see. For example, poly A tail bound with poly A tail binding protein interacts with the proteins which are associated with the 5' end of the transcript, causing a circularization of the mRNA that promotes translation. Again I repeat, the poly A tail binding protein found on the poly A tail, it will interact with the proteins which are found on the 5' end of the mRNA and thereby causing circularization of the mRNA. Okay, now important thing about the miRNA. I have also said that 3' untranslated region also have the regulatory regions which will allow interaction to the regulatory protein, as well as it will allow interaction with the miRNA. Actually miRNA sequence have a, they are the short sequence and their sequence are complementary to that 3' untranslated region. That is the reason that they binds. The 3' untranslated region plays a crucial role in gene expression by influencing the localization, stability, export and translation efficiency of an mRNA. This 3' untranslated region, it plays a very important role in gene expression by influencing the localization of the mRNA, stability of the mRNA, export of the mRNA to the cytoplasm and the translation efficiency of mRNA. These 3' UTR have various sequences that are involved in gene expression, like it contains a sequence which involve in gene expression that you know. I have also say you one of the elements that are found in the 3' UTR before, that is your CPE element found on the Xenopus laevis, right? But it also contains some other more regulatory sequence like your microRNA response element or MRE, where your miRNA bind, AU rich element ARE, which I have said just now. Many UTR 3' UTR have AU rich elements. Proteins that bind to these AU rich elements affect the stability or decay rate of the transcripts. So they also contain AU rich elements, right? And poly A tail, right? So what I have said, I have said that it contains various sequence, okay, this 3' UTR which will involve in gene regulation, which will involve in gene expression also, like your MRE element. MRE element is actually the place where your microRNA or miRNA will bind and interacts and carries the regulation. The below figure will show you that how it regulates, okay. Then I have said that it contains various sequence, which are involved in gene expression, okay, like your miRNA response that I have said just now, AU rich element that is ARE and poly A tail. In addition, the structural characteristics of 3' UTR as well as its use of alternative polyadenylation to play a role in gene expression. The structural characteristics of 3' UTR as well as its use of alternative polyadenylation to play a role in gene expression. So here we can see the miRNA. The miRNA, here it is showing a 5' cap it has not been showing clearly. If you can able to recognize this 5 prime cap of miRNA and a poly A tail. So in the left, extreme left, you can see, and it is occurring within the nucleus. So this 5 prime cap on the 5 prime inner poly A tail, so it is processing to form that miRNA, right? To form that miRNA, and this miRNA is further processed into single stranded miRNA by the help of dicer enzymes. Okay, and this miRNA binds complementary to the region of 3 prime UTR. It binds complementary to the 3 prime UTR, okay, with the help of another enzyme, with the help of another enzyme complex known as RISC, ribosome induced silencing complex, and it binds. This actually triggers that enzyme to bind to that site of the mRNA. What enzyme? RISC. RISC binds with this miRNA and goes to that place where it has to be regulated, the mRNA which has to be regulated. With the help of this miRNA sequence, it binds to the target mRNA on the 3 prime UTR, and then RISC will do their main function to cleave and degrade that mRNA. Okay, sometimes it also acts as an inhibitor. miRNA also acts as an inhibitor to the translational machinery by blocking the pathway of the ribosome, okay, thereby prematurely regulating the translation. So normal pathway of miRNA and siRNA that I will discuss you later on, the short interfering RNA is same. Here the enzymes has not been given, which helps that this miRNA to process. One of the enzyme is the dicer enzyme. It will cleave that double stranded miRNA into single stranded miRNA, as you see here, okay, as you can see here. See the miRNA duplex and mature mRNA. For this to follow this step, it has to process through dicer enzymes, right? It has to process through dicer enzymes, and then this miRNA does not bind alone. It has to be associated with your RISC, ribosome induced silencing complex, which will then helps them to bind to the target mRNA, which has to be destined for degradation or regulation. miRNA, with the help of miRNA, the RISC can able to bind to the target mRNA, because miRNA have a sequence which is complementary to the 3 prime UTR. I have said na the mRE element just now, the mRE elements, microRNA response elements, right? And then it can trigger the with the help of RISC, now it can trigger the degradation of that mRNA. Sometimes miRNA also acts as a blocker for the translational machinery to begin translation. Okay, so microRNA response elements, what they are saying here, let us see. The 3 prime, I have said na mRE element, okay, so the 3 prime UTR often contain microRNA response elements where your miRNA will bind. I have just now said to follow the miRNA pathway. So the 3 prime UTR often contains a microRNA response elements which are sequence through which your miRNA will bind. miRNA are short non-coding RNA molecules which bind to the mRNA transcripts and regulating the expression of that miRNA, regulating the expression of mRNA. Again I repeat, miRNA are what? They are short, they are non-coding RNA which are capable, they are short single stranded non-coding RNA molecule and capable of binding to mRNA transcripts and regulating the expression of that mRNA. How it can done? it cannot done alone. With the help of some proteins like your dicer enzyme, like your RISC, mainly ribosome induced silencing complex, it associate with that complex, and then it binds to the target mRNA by complementary base pairing, and then it can able to regulate the expression of that mRNA. Once miRNA mechanism involve partial base pairing of the 5' sequence of an miRNA with the mRE element, this binding then cause translational repression. 5' seed sequence, it refers to that a, when we cut that when we process that miRNA have a 5' in and a 3' in, right? So they interacts with the help of RISC. It goes and interacts with the target mRNA on the mRE element, which is located on the 3' UTR, and it binds by complementary base pairing. So RISC also able to interact with that mRNA, target mRNA, and then it will induce your regulation by degrading that mRNA. Okay, because RISC also have a hydrolyte, it also have a exonuclease activity actually. Okay, then comes your AU rich elements. I have also said that miRNA also contain AU rich elements. In addition to the MRE, the 3' UTR also contain AU rich elements called ARE. These AU rich elements are about 50 to 150 base pair long and usually contain many sequence of A and U. And these are the sequence A, triple U, A. Now within these AU rich elements, ARE binding proteins bind to these AU rich elements. ARE binding protein binds to these AU rich elements, which is dependent on tissue type, cell type, timing, cellular localization, and environment. So AU rich regions, the AU rich elements of the 3' UTR, the AU rich elements which are found in 3' UTR, they are your AU rich element binding protein binds, and it binds in a manner which is dependent upon tissue type. What type of tissue it is, depending on that it will bind, the cell type, the timing, cellular localization, and environment. In response to different intracellular and extracellular signals, these AU rich element binding protein can promote your mRNA decay. It can also affect the mRNA stability. So this AU rich element binding protein actually affect your mRNA stability. It will activate the translation also. This mechanism of gene regulation is involved in cell growth, cellular differentiation, and adaptation to external stimuli. So which type of tissues will be having activation of the gene, which cell type will be now growing and dividing. That means from the embryonic stage, when it grows, these AU rich regions, AU rich elements that are found on 3' UTR play an important role, where your AU rich element binding protein binds, and it in response to signaling, in response to extracellular intracellular signaling, this AU rich element binding protein, which is called AREBPs, can promote your mRNA decay. It promotes the effect of mRNA stability. It will also promote or activate the translation. And this mechanism of gene regulation involved in cell growth, it involves in cell differentiation and adaptation of external stimuli. Okay, and that is the main reason it has to act on some stimuli, some signals. Okay, some extracellular or intracellular signals like your cytokines, like your growth factors, which will act as a signals for allowing this ARE, that is AU rich element binding protein, that interacts with this AU rich element, ARE element found on the 3' UTR, to allow expression, to allow regulation of the gene. Okay, to allow the translation also. Okay, so these are the external stimuli. These are the some signaling signals. They act as signals like cytokines, growth factors, tumor suppressors, proto-oncogenes, cyclins, enzymes, transcription factors, receptors, and membrane proteins. So on the basis of this cytokines, growth factors, tumor suppressors, proto-oncogenes, cyclins, enzymes, transcription factors, receptors, and membrane protein, this AU-rich binding protein can promote the mRNA decay, can actually, better to say, can regulate the transcription of that particular gene, can regulate the expression of that particular gene, expression of that particular mRNA of that gene, better to say mRNA. Okay, so this is about the AU rich elements. So you can see it is a vast topic. It cannot be ended. You cannot end your discussion based on this regulation. Now comes the circularization of mRNA that I have already discussed in the 5' UTR. That circularization of mRNA transcript is mediated by proteins interacting with the 5' cap and poly A tail. So you can see here clearly EIF4E, EIF4A, and EIF4G. They interacts with the 5' cap, whereas the poly A tail binding protein, as written as PABP, bind to the tail, bind with the poly A tail. Now the proteins which are found on the 5' cap interacts with the poly A tail to form circular mRNA. So poly A tail contains the binding sites for poly A tail binding protein, as I have said. It is the binding site. The poly A tail is the binding site for the poly A tail binding protein. These poly A tail binding protein cooperate with other factors, the factors which are found in the 5' site, or it can also cooperate with the other proteins also, masking, right? So these poly A tail binding proteins cooperate with the other factors to affect the export, stability, decay, and translation of mRNA. PABP is found in the poly A tail, which may also interact with the proteins like translation initiation factors that are bound to the 5' cap of the mRNA. This interaction, so I have said that PABP, this poly A tail binding protein, bind to the poly A tail, and this poly A tail binding protein can also interact with other proteins, like it can interact with the translation initiation factors, as you can see here. This EF4E, EF4G, EIF4E, EIF4G, EIF4A are what? They are the transcription factors. They are the initiation factors of translation. Sorry, not transcription, translation initiation factors, right? So it can interact with the translation initiation factors, these poly A tail binding protein, which are bound to the 5' cap of mRNA, and that cause circularization of the transcript, right? This interaction, by interaction of the 5' cap associated by the initiation factors of translation, it interacts with the poly A tail binding protein that cause circularization of the transcript, which subsequently promotes translation initiation. Furthermore, it allows efficient translation by cause recycling of the ribosomes, as I have said in the audio lectures in WhatsApp. I have said that ribosome recycling also occurs by this circularization of the mRNA, so that repeatedly more rounds of translation occurs in that same mRNA. While the presence of poly A tail helps in triggering translation, absence or removal of poly A tail leads to exonuclease mediated degradation of the mRNA. If the poly A tail is not present, the poly A tail the poly A binding site, the poly A sequence also found within the 3' UTR, that leads to the formation of poly A tail as done by poly A polymerase. Okay, so if there is no tail, or removal of this tail, then it will trigger the exonuclease activity to degrade that mRNA, right? So polyadenylation itself is regulated by sequence of 3' UTR of the transcript. As I have stated before, the CPE elements, the cytoplasmic polyadenylation element binding protein. It is highly conserved RNA binding protein. So it is what they are saying. Let us see. Polyadenylation itself is regulated by sequence within the 3' UTR of the transcript. The polyadenylation is regulated by the 3' UTR. Within the 3' UTR, there are some sequence where some regulatory proteins bind and regulates the polyadenylation process. This sequence includes the CPE, cytoplasmic polyadenylation element binding protein, which is a uridine rich sequence that contribute to the polyadenylation activation and repression. So the CPE means cytoplasmic polyadenylation element. This cytoplasmic polyadenylation element found on 3' UTR, and within this cytoplasmic polyadenylation element your CPEB protein binds. As I have mentioned to you in the beginning, the CPEB, that is cytoplasmic polyadenylation element binding proteins, bind to the CPE elements of the 3' UTR. Okay, the CPE are uridine rich sequence, fully consist of uridine, that will contribute to polyadenylation activation as well as repression. The CPEB binding protein, that is a cytoplasmic polyadenylation binding protein, binds to the CPE in conjunction with a variety of other proteins for providing various type of responses. So this is about the 3' UTR. In the next lecture, I will talk you about the role of especially about the siRNA, the miRNA. The pathway of miRNA is also similar with the siRNA.